everyone, and welcome to today's webinar, Unraveling the Potential of Neural Stem Cells, Antibodies as Key Tools. I'm Christy Jewell of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, you can easily click on that Thermo Fisher logo or visit thermofisher.com. So let's get started. We encourage you to participate today by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. You may also use the Ask a Question box to let us know if you're having any trouble seeing or hearing the presentation. I would now like to welcome our speaker for today's webinar, Jamuna KS, Scientist 2, Cell Biology, Thermo Fisher Scientific. For a complete biography on Jamuna, please visit the Biography tab at the top right of your screen. Jamuna, welcome. You may now begin your presentation.
Thank you for that very nice introduction. And hello, everyone. The title of today's presentation is Unraveling the Potential of Neural Stem Cells, Antibodies as Key Tools. And over here, we'll be discussing 4 topics. A, background of NSC research and neurogenesis. B, stem cells, pluripotent stem cells, and neural stem cells. We'll discuss about differentiations from stem cells and their characterization. We'll also be talking about disease modeling with focus on Parkinson's disease. We'll get started with a brief history of neural stem cell research. NSCs were discovered in rat brain in the year 1960. Until then, the dominant opinion in the scientific community was that in the mammalian central nervous system, the ability to generate new cells or neurogenesis was arrested after birth. This theory was proven wrong after the discovery of NSCs and neurogenesis, and since then a great amount of research is focused on understanding NSCs for the development of cell therapy in order to treat central nervous system diseases. Now, why is it so important to study NSCs? The reasons are that they help in understanding development, disease progression, or neural tissue regeneration. They aid in optimizing stem cell-based treatment of neurological disorders or injuries. And now that we are able to isolate and analyze the neural progenitor populations in vitro, they have been extremely important in deciphering the cellular and molecular mechanism underlying neurogenesis. Coming to the clinical application of NSCs, the human neurological disorders like Parkinson's, Huntington's, ALS, Alzheimer's disease, or stroke are all caused by the loss of neurons and glial cells in the brain or the spinal cord. So for all of these disorders, NSCs are promising tools towards cell replacement therapies. Moving on, the ability of brain to create new neurons and connections between those neurons is called neurogenesis. Neurogenesis occurs through the division of neural stem cells and subsequent maturation into neural progenitor cells. And these progenitor cells migrate and mature to form neurons. NSCs, if you see, NSCs can generate into all neural cell types. They are most active during prenatal development, but they continue to divide throughout the adult life. NSCs are found predominantly in the subventricular zone of lateral ventricles and subgranular zone of hippocampus. While talking about neurogenesis, it's important to highlight on the prefrontal cortex because this region connects to most parts of the cortical and subcortical brain. And this is a region which is involved in a number of neural networks, and it's susceptible to damage, which makes it very important in neurodegenerative diseases. The PFC or the prefrontal cortex is the part of the cerebral cortex covering the frontal lobe. This is also the region which is the last to develop. So if you see, the brain develops in a back to front pattern and the prefrontal cortex develops in the end. This region takes care of decision making, personality expression, and it also moderates the social behavior. An important point to mention over here is that the stem cell pool or the neuroplasticity reduces with age. So neuroplasticity is the ability of the brain to adapt to change, and this, along with the stem cell pool, declines over time. So with this, we have covered the brief background of NSCs. And coming to the next part, which is stem cells, they have transformed both science and medicine due to their biological properties and the potential medical importance. The major biological properties which distinguish stem cells from any other type of cells include A, asymmetric cell division. Now, an asymmetric cell division is defined as any division which gives rise to two daughter cells of different fates. Right? So this is very important in maintaining tissue homeostasis because one daughter cell retains the ability of self-renewal and pluripotency, whereas the other enters the path of differentiation. The other distinguishing properties are self-renewal and pluripotency. These cells are also capable of existing in a mitotically quiescent form. Now, what is this mitotically quiescent form? So this is a stage where the stem cells remain out of cell cycle unless there is a requirement. This stage is critical to ensure lifelong tissue maintenance and to protect the stem cell pool from premature exhaustion under the stressful conditions. The medical importance of stem cells include, they help in enhancing our basic knowledge on disease and development. They are also helpful in drug discovery and in cell-based therapies. These cells, because of their ability to differentiate into any other cell types, they have enabled the study of primary and difficult to obtain cell types, which would otherwise be inaccessible for research due to their rare tissue origin or ethical constraints. And also, since these stem cells can be used to generate physiologically relevant models, they have excellent potential to replace animal studies. Here in this presentation, we'll be discussing two types of stem cells, NSCs and PSCs, or the pluripotent stem cells. Now, NSCs or the neural stem cells are the stem cells of the nervous system. They are present in developing an adult mammalian central nervous system. These cells serve as excellent models for research focused on neural development and neurological disorders. They are also used to develop potential treatments for incurable neurodegenerative diseases and neuronal injuries. Now, talking about the sources of NSCs, they're found in the mammalian brain, they're found in the subventricular zone and the dentate gyrus of hippocampus. They can also be derived from embryonic stem cells and the induced pluripotent stem cells. Here in this representation, we can see that pluripotent stem cells give rise to multipotent neural stem cells. These multipotent stem cells give rise to lineage-restricted cells, the glial progenitors and the neuronal progenitors, which can further differentiate into astrocytes, oligodendrocytes, and the neurons. Next, we'll discuss a bit about PSC or the pluripotent stem cells. The commonly studied PSCs are embryonic stem cells and the induced pluripotent stem cells. If you have a look at the first diagram, so after the 32-celled morula stage, the blastocyst forms, which is roughly 64 to 200-celled stage. Blastocyst has trophoblast cells in the outer and the inner cell mass cells towards the interior. These inner mass cells can be isolated and propagated in culture. They are termed as embryonic stem cells or the ESCs. Now, iPSCs can be generated from a healthy or a deceased individual by the ectopic expression of reprogramming genes such as OCT4, SOX2, c-MYC, and KLF4. This can be done in adult somatic cells. For example, the skin fibroblasts are the cells which has been extensively studied for reprogramming. both ESCs and iPSCs maintain the properties of pluripotency and self-renewal. Now that we have discussed about stem cells, we'll elaborate more on their application part. The major applications of stem cells are in developmental studies, disease modeling and drug discovery, and regenerative medicine. Stem cells are the natural units of embryonic development and tissue regeneration because they can generate multiple cell types. And due to this, they help in understanding the processes that transform a single cell into a multicellular organism. For example, one example is that of the central nervous system. The central nervous system, especially the brain, is very sensitive to various chemical substances and to physical factors. So in this case, stem cells can be used as excellent model to study developmental neurotoxicity. The second figure is a representation of disease modeling. From a patient, fibroblasts can be isolated, and these fibroblasts can be reprogrammed into iPSCs. These iPSCs can be differentiated into any cell type which would mimic the disease condition. The differentiated models can be further used for patient-specific therapy. The next major application of stem cell is in regenerative medicine, wherein it can be used to replace the damaged cells or to regenerate organs. The figure shown over here is from a case report on transplantation of human-induced pluripotent stem cell-derived cardiomyocyte patches for ischemic cardiomyopathy. Here, cardiomyocyte patches were generated from allogenic iPSCs and were transplanted into a patient through thoracotomy. The clinical symptoms improved 6 months after the surgery without any adverse events, suggesting that the patches were well tolerated. Until now, we discussed on the introduction to stem cells and their applications. Now we'll focus on differentiations and characterizations. Differentiations of PSCs into specialized cell types. PSCs have the ability to differentiate into cell types of the three embryonic germ layers, the ectoderm, mesoderm, and the endoderm. Ectoderm is the external layer which gives rise to skin, neural, and the pigment cells. Mesoderm is the middle layer which gives rise to kidney, cardiac, skeletal muscle cells. And endoderm is the internal layer which gives rise to liver, lung, pancreatic cells, etc. Now, in the ICC panel, we have used the significant marker of each stage in order to characterize it. So if you see, we have used beta-3 tubulin to characterize ectoderm. Smooth muscle actin or SMA has been used to characterize the mesoderm stage, and alpha fetoprotein has been used to characterize the endoderm stage. So why is characterization important, especially in stem cell culture? The long-term culture or genomic editing affects the quality of stem cells. Therefore, we need to characterize these cells in order to assess their quality, in order to check if their fundamental properties are intact. Characterization helps in identifying the differentiated cell types. It also helps in checking if the differentiation is proceeding in the right direction. So when you're performing a differentiation from PSC to a specific cell type, the differentiation happens through multiple stages. So it's important to check for markers of each stage in order to ensure that your experiment is proceeding in the right direction. Characterization is also helpful in detecting the markers of self-renewal and pluripotency, which are the major characteristic properties of stem cells. Now, considering all of these, there's a critical need for extensively tested and specific antibodies for stem cell markers. Now, how do we know if our antibodies are well tested? How do we check if it works in the application that we are planning to perform? So we at Thermo Fisher have certain testing approaches for advanced verification of our antibodies, which builds confidence in selecting the right antibody for the intended research need. Over here, multiple strategies are employed to verify target specificity, and all our antibodies are tested using at least one of these specificity verification methods. The specificity verification methods which we employ over here are knockdown, cell treatment, and relative expression. So in knockout, the gene for a particular protein is silenced through CRISPR-Cas9 technology. In knockdown, the gene of interest is knocked down using siRNA technology. Using cell treatment, we detect the downstream events following a cell treatment. And in relative expression, we use the naturally occurring variable expression of proteins to confirm specificity. To explain a bit on this, if we have an antibody against a particular protein, we pick a cell line which highly expresses the protein. We then perform knockdown or knockout of the gene encoding the protein in order to ensure the specificity of the antibody. This specificity testing combined with the application validation ensures that the antibody works in the right application of interest. The applications which we test our antibodies are flow cytometry, Western blotting, CHIP, IHC, and ICC. On our website, we have something called the Advanced Validation Badge, which helps the customers identify antibodies which have undergone rigorous specificity testing. This badge can be found in the search results and at the top of product-specific web pages. Data supporting the Advanced Verification Badge can be found in product-specific data galleries. Over here, if you look at the P53 monoclonal antibody, you can see this advanced verification tag. There are 15 published figures for this antibody along with the 166 references. So all these are parameters which help in selecting a good antibody. Now that we have seen how to pick a good antibody, let's have a look at some characterization data. The significant transcription factors of pluripotent stem cells are OCT4, SOX2, NANOG, and Link28. The major cell surface proteins are TRA160, TRA181, SSEA3, and SSEA4. All these factors are important in regulating cell division, differentiation, and development. The bright field image shown over here is that of a typical iPSC colony, and we have characterized it using the significant pluripotency factor OCT4. And if you see, this OCT4 antibody is very specific and it shows a very good nuclear staining. We have also checked the expression of other factors like NANOG, Link28, and SOX2. So this characterization data ensures that our stem cells are of good quality. In the coming few slides, we'll discuss about few differentiations from the ectoderm lineage. As what we have mentioned earlier, ectoderm is the outermost germ layer, which is the precursor of skin, nervous system, etc. The first differentiation that we are going to focus on is pluripotent stem cell to Schwann cell progenitors. Schwann cells play a crucial role in successful nerve repair and regeneration. They're also used to study myelin repair and nerve impulse transmission. The differentiation proceeded in two stages. The first stage is PSC to neural rosettes, and the second is neural rosettes to Schwann cell progenitors. For the first stage, the basal media used was a combination of knockout DMEM, F12, and neurobasal. Into this, we added N2, B27. We also added a GSK3 inhibitor, which is CT99021, and a TGF-beta inhibitor, which is SB431542 compound. PSCs were grown in this media for 6 days in order to generate neural rosettes. In the next stage, along with the differentiation media 1, a high concentration of NRG1 was added. NRG1 or neuregulin is a Schwann cell lineage promoting factor. This was continued for 18 days after neural rosettes to generate expandable human Schwann cell progenitor cells. Now that the differentiation steps are over, how do we actually understand if our cells have got differentiated? For that, we have performed characterization using the antibody SOXTIN. The reason for picking SOXTIN is because it plays a crucial role in Schwann cell development. Now, in this ICC panel, you can see that we have checked the expression of SOXTIN in control cells and differentiated cells. Control is the undifferentiated iPSCs. So here we can you can see that the expression of SOXTIN is absent in the control cells, whereas it is present in the differentiated population. This antibody-based characterization helps to ensure that our cells have got differentiated into Schwann cell progenitors. The next differentiation is PSC to retinal ganglion cells. These cells would be helpful in glaucoma research, in the study of optic neuropathies and demyelination disease. Over here, we have followed a 28-day protocol wherein PSCs were grown in suspension in a spheroid form for 4 days in the RGC media 1, followed by plating them and growing until day 28 in the RGC media 2. The basal media used is DMEM F12 and it is supplemented with glutamax, knockout serum replacement, B27, Noggin, which is a BMP4 inhibitor, a Wnt inhibitor DKK1, and IGF-1, which is an AKT activator. All these are components of RGC media 1, and the RGC media 2 contains all the components present in RGC media 1 along with BFGF. Now, the characterization of this differentiation was performed using very specific antibodies against optineurin and beta-3 tubulin. The reason to choose these antibodies are that optineurin is highly expressed in RGCs and beta-3 tubulin is a neuronal marker. In this ICC panel, you can see that the expression of optineurin and beta-3 tubulin are absent in the control cells, whereas they are highly expressed in the differentiated cells. So these two antibodies have helped us in understanding that our PSCs have got differentiated into retinal ganglion cells. Next, we are going to talk about neuroepithelial cells and the neural rosettes. Both these are stem cells of the central nervous system from which neurons and glia are formed. These cells are used to study neurogenesis. Neuroepithelial cells were generated by growing PSCs in our in-house developed supplement for 6 to 7 days. These cells were characterized using the antibody OTX2, since OTX2 plays a major role in brain development. Even here, the expression of OTX2 was compared with the control PSCs, and it's absent in control, whereas the expression can be clearly seen in the differentiated cells. Next differentiation from PSC is to neural rosettes. We have already discussed about this protocol during that of Schwann cell progenitors. Here we have characterized neural rosettes using nestin, which is a neuronal marker. The nestin antibody is highly specific, as what you can see from the ICC panel. There is no expression in the control PSCs, whereas the antibody has stained the differentiated neural rosette population. So while performing the Schwann cell differentiation, one stage that comes in between is that of neural rosettes. Performing characterization in this stage ensures that our characterization is proceeding in the right direction. So this actually acts like a checkpoint of differentiation. The next differentiation that we'll be discussing is PSC to cerebral organoid. This model is very helpful in studying the complexity of brain, studying the roots of autism, for that matter, any neurological disorder. We have followed the traditional Lancaster protocol for this differentiation, which continued for more than 50 days. The major steps were germ layer differentiation, neural ectoderm induction, neuroepithelial bud expansion, and brain tissue growth and expansion. After the neural ectoderm stage, the developing organoids were embedded in matricial rocks and grown in suspension in an orbital shaker for more than 30 days. The previous differentiations which we discussed were all in 2D, where a cerebral organoid is a 3D structure which was approximately 5 millimeter in size. For 2D monolayer differentiations, we followed the regular ICC steps, but it was little different for the 3D models. For cerebral organoid, the fixation was performed for 30 minutes, followed by dehydration in 30% sucrose solution overnight. After the dehydration step, the organoids were sectioned. All our sections are of 10 micron thickness. Now, post-sectioning, the ICC protocols were similar to that of what we do for 2D. So over here, you can see that in the ICC panel, we have performed characterization using very specific antibodies against beta-3 tubulin, SOX2, Frizzle9, and Vimentin. Beta-3 tubulin antibody has stained the neurons. SOX2 antibody has specifically stained the NSEs. Frizzle9 is a hippocampal marker, and Mymintin antibody has stained the cells of radial glia. Now, in the following two slides, we'll be discussing about NSEs and differentiations from NSEs. NSEs are very important cell types for neuroscience research. They help in developing cell therapies to treat CNS injuries and diseases. The bright field image which you show over here is how NSEs look under microscope. Here we have characterized NSEs using antibodies Nestin and SOX2. Both these are the major markers of NSE, and both of these are very good antibodies as what you can see from the ICC data. Coming to the culturing part, NSE culture and differentiation was performed according to the protocols mentioned in the GIBCO NSE handbook. The media used to grow NSEs are knockout DMEM F12 plus glutamax, along with stem proneural supplement and EGF and BFGF. Stem proneural supplement not only supports the expansion of neuronal progenitor cells, it also enables the cells to retain their progenitor status and the potential to differentiate into neuronal and the glial cells. Coming to differentiations from NSE, first one is NSE to neurons. Now, how do we do this? We do this by adding neurobasal media supplemented with B27 and glutamax into the NSEs. Neuronal differentiation can be observed within 7 days of addition of differentiation media. And over here, the characterization of neurons were done using specific markers, beta-3 tubulin and MAP2. The next differentiation is NSE to astrocytes. This differentiation was performed by adding the media consisting of DMEM, N2, FBS, and glutamax into NSEs. Astrocytes can be seen within 14 days of differentiation. We have characterized astrocytes using antibodies against S100 beta and GFAP. S100 beta is a calcium binding protein which is found abundantly in astrocytes, and GFAP is a glial fibrillary acidic protein which is a hallmark intermediary filament protein found in astrocytes. Both these are very specific antibodies which help in distinguishing the differentiated population. That's all about differentiation. So until now, we have seen about stem cells and the differentiations from them. Now we'll see how these cells and the differentiations from them can be used for disease modeling. And the disease that we are going to focus on is Parkinson's disease. Parkinson's is a neurodegenerative disease which results in the loss of... Parkinson's is a neurodegenerative disease caused by the loss of dopaminergic neurons in the substantia nigra, and this reduction of dopamine results in the impaired control of movements. PSC-derived DA neurons are excellent models to study Parkinson's disease. DA neurons can be characterized easily using specific markers FOXC2 and tyrosine hydroxylase. They can be used to study disease-associated genes like PARC2, PARC7, PINK1 or SNCA. In order to study Parkinson's disease, there's a need of highly specific antibodies against key proteins associated with the progression of Parkinson's disease. Now, how do we use PSCs in Parkinson's disease? This workflow explains it. So here, iPSCs were differentiated into floor plate progenitor cells. This is a stage that comes before DA neurons. The floor plate progenitor cells were characterized and sorted. The sorted cells were differentiated into DA neuron progenitors. These cells were transplanted into the striatum of the brain. It was in the year 2018 that the first human trial for Parkinson's disease iPSC transplantation began in Japan. The publications mentioned over here are all about using stem cells in treatment or studies of Parkinson's. I'd like to specifically call out the second article mentioned over here. which is the treatment of Parkinson's disease through personalized medicine and induced pluripotent stem cells. This was published in Cells in the year 2019, and it highlights multiple clinical trials for neurodegeneration using stem cells conducted worldwide. In this slide, we'll see how to develop a model for Parkinson's disease. If you have a healthy iPSC line, generate Cas9 iPSC out of it. This Cas9 iPSC can be used to edit the LRRK2 gene. Why LRRK2 gene? That's because mutations in the LRRK2 gene is the major factor contributing to the genetic development of Parkinson's disease. After the mutation is introduced, the clone is isolated and expanded. This can be done in the routine stem cell media. Over here, we have used stem cell media along with the Revitacell supplement. After obtaining the right clone, they can be differentiated into DA neurons using PSC dopaminergic neuron differentiation kit. We'll be discussing more about this kit in the coming slides. Once the DA neurons are generated, they can be characterized using specific antibodies, FOXA2 and tyrosine hydroxylase. FOXA2 regulates midbrain DA neuron specification, and tyrosine hydroxylase converts tyrosine into dihydroxyphenylalanine, which is converted into dopamine. The data you see over here is of DA neurons differentiated from healthy iPSCs. Coming to this case study over here, so here they have differentiated Cas9 iPSC and LRRK2 mutated iPSC into DA neurons. They've characterized them using tyrosine hydroxylase as well as beta-3 tubulin antibody. And if you see the images, the differentiations from both Cas9 iPSC as well as the LRRK2 mutant look very similar, which means that DA neurons were successfully generated from both of them. Now, these are the kits that can be used for DA neuron differentiation and characterization. The PSC dopaminergic neuron differentiation kit can be used for the generation of DA neurons within 35 days. This kit is a 3-step system. It has one basal media called the floor plate cell expansion base and 3 supplements each for floor plate specification, floor plate expansion, and DA neuron maturation. Within 10 days in the floor plate specification media, floor plate progenitors are formed. They can be expanded in the floor plate expansion media. These expanded cells can be banked as well, so that next time you do the differentiation, you can save these 16 days. The floor plate progenitor cells can be plated and matured to form DA neurons in the maturation media. So using this kit, we can differentiate PSCs into DA neurons in 35 days. We also have a characterization kit for DA neurons. This kit contains the 3 antibodies, OTX2, FOXA2, and tyrosine hydroxylase, along with other reagents used to perform immunocytochemistry. OTX2 and FOXA2 are markers for intermediary floor plate progenitors, and tyrosine hydroxylase is a marker for mature DA neurons. Coming back to the case study, over here they have studied the neuronal outgrowth in Cas9 iPSC as well as the LRRK2 mutated iPSC line. They've also considered the homozygous and heterozygous mutation of A30P, also known as alpha-synuclein gene. This is another gene associated with Parkinson's disease. Here they observed that in LRRK2 mutants, the DA neurons showed a delayed outgrowth after replating. But even though the onset of neurites were delayed, the DA neurons from this resulted in longer neurites. Further, they tried to study the sensitivity of DA neurons from Parkinson's disease towards neurotoxins. For this study, the control cells as well as the LRRK2 mutated cells were treated with DMSO, staurosporin, and 6-hydroxydopamine. Staurosporin is an apoptotic inducer, and 6-hydroxydopa is a compound used by researchers to destroy DA neurons in the brain. High content analysis reveals that LRRK2 mutated DA neurons were more sensitive to 6-hydroxydopamine than the control cells under normal glucose conditions. When these cells were further stressed by the removal of glucose, the effect of 6-hydroxydopa was more pronounced as it manifests in SNCA30P homozygous neurons as well. So with this data, we see that Parkinson's DA neurons were more sensitive to neurotoxins than the control cell lines. That's all about disease modeling. Now we'll focus on the recent researches for the use of NSE in therapy. The first paper that we'll be discussing is human-induced neural stem cells support functional recovery in spinal cord injury models. Spinal cord injury, or SCI, is a condition that leads to permanent and progressive disability in sensory or motor functions. Unfortunately, there's nothing that can reverse the damage. So here, they have generated induced neural spinal cord injury. The first paper that we are going to discuss is human-induced neural stem cells support functional recovery in spinal cord injury models. Spinal cord injury, or SCI, is a condition that leads to permanent or progressive disability in sensory or motor functions. Unfortunately, there's nothing that can reverse the damage. Here they have generated induced neural stem cells from human urine cells and transplanted into injured spinal cord of rat. These NSEs survived, engrafted, and expressed the neuronal and glial markers. Large number of axons extended from the grafts over long distances, leading connections between the host and the graft neurons at 8 weeks post-implantation, with significant improvement in the locomotor functions. In this study, they've characterized the induced NSEs using antibodies against specific NSE markers like Nestin, SOX1, SOX2, and PAX6. I've not added the ICC data over here. They've also checked the expression of TUJ1, MAP2, along with GFAP, MBP, and neurofilament 200 in the differentiated induced NSEs at the transplant site. These antibodies have helped in identifying the NSEs as well as the differentiated NSEs in the model studied over here. The next article is extracellular vesicles derived from neural stem cells, astrocytes, and microglia as therapeutics for easing TBI-induced brain dysfunction. Here they have generated a traumatic brain injury model. After the injury, through the tail of the rat, NSE-derived and astrocyte-derived vesicles were injected. This resulted in improved motor function with higher expression of VEGFR2 and migration of NSEs and neuroblasts to the lesion site. These extracellular vesicles can be characterized using antibodies against specific markers like CD9, CD63, CD81, etc. Both these studies prove that NSEs have great potential towards treating the disorders which were once considered incurable, and antibodies play a major role in harnessing their potential. So far, we have discussed a lot about antibodies as the characterization tool. It is also important to highlight the role of instruments in this process. The nice images that I have shared with you are all acquired with our in-house microscopes and platforms. Whether you're looking at 2D monolayer or 3D neurosphere samples that require qualitative or quantitative analysis, we, Thermo Fisher, definitely have a product solution that fits your needs. Highlighted here are our EVOS microscopes and high-content screening platforms, both capable of providing beautiful, high-quality images for publication process. And if you want to quantify fluorescent signals from the entire well or various plate formats, Varioscan Lux Plate Reader is a good option for you. Coming to the conclusion part, stem cells show tremendous potential in the areas of developmental biology, disease modeling research, drug development screening, and cell therapy studies. Validating PSCs and their progeny to confirm pluripotency or ensure differentiation into the desired cell type is critical for stem cell research. Antibody-based detection methods such as ICC and flow cytometry are the commonly used characterization methods for stem cells and differentiated cells. The use of high-quality antibodies is one key factor contributing to the success and rapid progress of stem cell research. Over here, we have highlighted posters of antibodies for stem cell research and neuroscience research. We have also tagged the resources that would aid in stem cell research and NSC research. Yeah, that's all from my end. Thank you, and I'd be happy to take any questions if you have.
Jamuna, thank you so much for that informative presentation. It is now time for our live Q&A portion of our webinar. Now, to our audience, if you have any questions you would like to ask, please do so now. Just click on that Ask a Question box, type in your question, and click Submit, and we will answer as many of your questions as we have time for. Jamuna, we already have some great questions coming in. I am going to start with this one. Do we have different staining protocols for 2D versus 3D cells?
So the protocols for 2D and 3D staining are very much similar. The difference is that when it comes to 3D models, the incubation step, the duration of the incubation step at each stage of ICC would be longer. And this duration would depend upon the size of the organoid with which you are working. And also, because these organoids are large in size and they also contain water, proteins, and lipids in them, they need additional steps like, dehydration or the tissue clearance to be performed in order to remove all these additional factors. Because if these are there, they make your organoids, they make it not very clear for microscopy. So it's important to remove these additional components so that you will get good images. Yeah, so those are the differences. So the extra steps would be the dehydration as well as tissue clearance, and then the longer incubation steps. Yeah.
Thank you, Jamuna. Our next question, is there any specific consideration in selecting the type of antibodies used in the 3D staining?
As such, there are no specific consideration while selecting antibodies for 3D staining. But like what I mentioned before, you need to pick the antibodies which are very well validated. And also you have to ensure that the antibody is working in the application which you intend to work on. So these are the major considerations that need to be considered while picking the antibody.
Very good, thank you. Our next question. This question is a little long and it's two parts. So recent research suggests that mesenchymal stem cells, MSC, could be more effective for Parkinson's disease treatment. Can you elaborate a little bit more on the characterization of MSC, and are there any differences with the characterization of PSC?
That's a very good question. MSCs, yes, they are widely used in Parkinson's research these days because MSCs can be, easily derived from adipose tissue, bone marrow, dental pulp, or your peripheral blood. And these cells are already being widely used in, osteoarthritis and myocardial infarction. So they already have a record of, established clinical applications. So coming to the characterization part, MSCs can be characterized on the basis of the surface markers which they express, actually based on the presence and absence of certain surface markers. They are also characterized based on their differentiation potential. So if you see, MSCs express certain surface markers, and the presence of these markers differ based on the source from which they are derived. For example, in adipose-derived MSCs, CD13 is present on the surface, whereas CD14 is absent in adipose-derived MSCs. So you see this presence and absence of surface markers. That's one way of characterizing MSCs. And another one is their differentiation potential, wherein MSCs are capable of differentiating into adipocytes, osteocytes, and chondrocytes. So these two are the major characterizations which we do for MSCs, as what I'm aware of. Yeah.
Very good. Next question. Can I use a super resolution microscope for analysis of this disease?
Yes, these microscopes can be used to, understand your differentiated populations or the diseased population. So yes, you can use these super resolution microscopes to see the cell types. Yes.
Very good. And can you please tell us, are there stem cell specific antibodies for Drosophila models as well?
Stem cell, is the question about Drosophila stem cells or the stem cells specific to Drosophila?
Yes, our attendee is asking if there are stem cell specific antibodies for the Drosophila model.
So if you want to look at the stem cell markers, that would be consistent in different models. So if I'm right, the antibodies which we use for use against the, but I'm not sure if there are against Drosophila models. I have to check and get back.
Very good. Thank you. And I do want to let our audience know that we do have your emails for all of your questions that are submitted. So if there's any follow-up needed, Jamuna is able to email you. So, okay, let's see. We're getting so many questions in here. How about, how can nanotechnology support and enhance neural stem cell therapy for Parkinson's disease?
Question, can you please repeat the question?
I sure can. How can nanotechnology support and enhance neural stem cell therapy for Parkinson's disease?
I'm sorry, I don't think I have an answer for question because I'm not an expert in that field. So I am not aware of the nanotechnology part.
Thank you. How can we ensure that all cells in our culture have completed their differentiation?
So yes, like what I mentioned, so characterization is the only option with which you will be able to figure out if your cells have got differentiated into the cell type which you want to differentiate. So characterization is the tool with which you can do that.
Thank you. Is Parkinson's disease identified by image J, biological images?
Parkinson's disease, so one of the significant differences or changes that happen in Parkinson's disease is the shortening of the neurite length. So if you have the DA neurons, the Parkinson's, which carry the Parkinson's condition, the length of the neurites would be shorter compared to the regular neurite. So that would be one method in identifying the Parkinson's cell type.
And Jamuna, from your experience, could you please share your biggest challenge in the disease modeling workflow? Are there any steps to take note
on? Challenges. So yeah, there are always a chance of having different cell types in your population. So when you work with stem cells, there are always chances of random differentiation, yeah, there are chances of random differentiation as well as any kind of stress would cause the cells to differentiate. So you need to ensure that you're working with the right cell type. So you need to perform characterization from the initial stem cell, including the middle stages, and then you need to characterize the final differentiated model. So the differentiated cell types, yes, those are one challenge, and then ensuring that you perform characterization at each step would make sure that you have the right cell type to work with. Yeah.
Very good. Thank you, Jamuna. And it looks like some of our questions may need some additional follow-up. So I'd like to give you an opportunity to give any final comments to our audience.
So yes, the resources and the posters which I've tagged in this presentation, they are very helpful for your stem cell research or NSC research. So please have a look at it. Those will have a lot of information that would help in your study. So yeah, please take time to have a look at those resources.
Jamuna, thank you so much for your time today and for this lively Q&A session. I do want to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. And before we go, I do want to thank the audience for joining us today and for their interesting questions. Those questions we were not able to answer today, and additionally, those that are submitted during the on-demand period, they will be addressed by our speaker via the contact information that you provided at the time of registration. Today's webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So as we close, a survey will pop up on your screen, and we encourage you to participate, and your feedback is greatly appreciated. Thank you again for joining us, and Jamuna, thank you again for your time today. Have a great night, everyone. Bye-bye.